Hi, everybody. This is the Chapter 11C PyMOL mutagenesis video tutorials that you guys could use to help you navigate through the PyMOL program and also assist you in completing the post-lab write-up. All right, so I distributed the Chapter 11C mutagenesis tutorial, and then I also released last year's Chapter 11A PyMOL tutorial. And both of those, I made sure in the instructions to download PyMOL that they're up to date for this semester's instructions. So if you don't have PyMOL already installed in your computer for whatever reason, if you got a new computer between last semester and now, visit this website. It's pymol.org/EP. The username to access the website installation files is JUN2020. For the username and for the password, it's alpha helix, all one word, all lowercase. If you do still have the PyMOL program from last year and it's still installed and it's running, great. You don't need to download the new version of PyMOL. Basically the methods and the tools that we're going to be using is pretty much very common through all different versions of PyMOL. So you don't really need to update or do anything like that. You can use the tutorial is kind of pretty skimpy in the instructions. This was really designed to supplement the tutorial that I would give in person in our discussion sections, but of course that's no longer available to us. So what I'm doing is this video will stand in for the in-person discussion PyMOL tutorial that I would give for about like an hour that students could follow along with their computers. The great thing is that now this is video recorded. It's going to be uploaded on YouTube. And I'm also going to annotate the different points, the time points in the video, such that if you miss something, you can go backwards. Or if you are familiar with like the first 3 or 7 steps, you can just pretty much skip all of that and just go to a certain specific instruction that you're looking for. So for your assignment of your protein, I also released this other PDF of which, depending on what lab sections you have, the enzyme or protein that is assigned to you, and also those corresponding PDB codes, since there are several and multiple PDB codes for these specific proteins on the Protein Data Bank website. We just want to make sure that we give you a PDB code that works for your enzyme or protein that we've assigned to you. For those sections the students that are assigned in the B2 lab section, I've kind of been going through a few of the students' PyMOLs sessions that they've dropped in on for office hours, and they've shared like their screen with me, and they've chosen the glycerol ligand, which is perfectly fine. But that's when I look at this glycerol ligand, it's definitely not the glycerol that I've known for a while. Basically, it looks like a little spider shaped or insect shaped kind of thing, and that's not quite correct. So I'm going to look at this file and get back to all of the B2 students. And I'm not going to give you guys extra work to do. I'll just basically give you special instructions so that you can complete and finish your PyMOL post lab write-up. But the PDB codes and also the enzyme protein is what you will need to complete prompts 1 and 2 of your post lab. So if we go back to the post lab template for the Chapter 11C lab, record the alphanumeric PDB code for the enzyme protein that you have been assigned to. That is essentially going to be this third column here for whatever lab section that you're in. So just go ahead and simply put in that PDB code there. Record the name/enzyme that you have been assigned. That's essentially going to be the information that's going to be in the second row there. So if you're in the B3 section, you would put in lactate dehydrogenase for the answer for the second prompt. And then your PDB code for the first prompt would be 1LDG. If you've noticed, pretty much prompts 1 through 4 are very, very easy. You can complete them in about 10 minutes. They're just kind of give me points for this post lab, as I don't want to make it too difficult in our circumstances right now. All right, so for the third prompt, you're asked to visit the protein data bank site, rcsb.org, and then search your PDB code, and then choose a ligand that has an appropriate 2D diagram image, just basically so that you can see exactly what the structure of your ligand is and familiarize yourself with it before you actually start delving into the PyMOL program itself. And also a viewable ligand interaction page under the 3D interactions. If you did follow through the tutorials for chapter 11AB, you'll notice that those instructions are very out of date, and the PyMOL, or I should say, sorry, the PDB website has gone through a lot of renovations. So a lot of these, those instructions that I gave last semester are no longer applicable, but I'll show you what this new PDB website looks like. It doesn't look very different. It's just when it comes to these different things from the chapter 11 AB tutorial, it varies a little bit. I am going to use the PDB code and the enzyme from those tutorials, and that's GAPDH, glyceraldehyde 3-phosphate dehydrogenase. And the PDB code for that is 1IHY. And when I look for that, I find it right here at the very top of only one of two structures that come up. I click on 1IHY here, and I'm taken to the main page for GAPDH in complex with ADP ribose. So this first main part of the PDB website just basically gives you a lot of the information that is associated with the experiments in which it was crystallized and also deposited, the paper also too, in where this structure was crystallized by these researchers, and also any other pertinent information. In the section for macromolecules, you'll find a lot of important information in terms of not only the name of the protein or the enzyme, but also the number of chains that are associated with your PDB code and PDB file when you load it onto PyMOL. And chains basically refers to the polypeptide chains that were crystallized and resolved using X-ray crystallography. And so in particular, when you load this PDB code onto PyMOL, you are going to essentially get what's going to be a homotetramer with chains A, B, C, and D. You're not going to be looking at all four chains. There's going to be a little trick that I'm going to show you to kind of hone in specifically on one chain and only work on one chain on PyMOL. And then a little lower, you're going to get into the small molecules section. Now, for GAPDH, the ligand APR as well as some sulfate ions were co-crystallized and resolved using X-ray crystallography for this particular type of experiment. We don't want students to use ions or metals or inorganic compounds such as sulfate to use as your ligand. We want you to use complex carbon-based molecules such as ATP, NADH, ADP, ribose, and so on and so forth. So I'm going to choose for my ligand for prompt C3. It says record both the ligand ID and the ligand name that you have selected. So I will choose APR as the ligand ID. So I will just put APR for my ligand ID. And for the name, luckily enough, I've gotten something that's just called adenosine 5-diphosphoribose. Some students have been picking ligands in which they have more complicated types of names. It's like 5-methyl-2-poly tert-butyl something. Don't worry about those complicated names. You don't need to refer to your ligand continuously as that complicated name. You can just say, the ligand APR or the ligand CEL or the ligand DZB or whatever. So when you finish that, we're pretty much just gonna now look at the two different pieces of data and information that were given here in the small molecule section. So for the 2D diagram, if we click on that, a image will pop up just showing what the structure is. And everything looks good. We have the adenine molecule here, and the ribose together makes adenosine, covalently bound to the diphosphates here, and then also finally at the end another ribose sugar attached there at the end of the diphosphate molecules. Here on 3D interactions, these have been now they were replacing the 2D interaction diagrams that some of you from the first semester of biochem are familiar with looking at. It kind of just had this like 2D structure of the ligand itself with hydrogen bond dash lines to other amino acid residues. But now it's replaced by this 3D diagram. And so when we click on it, we're brought to a new window in which kind of get this interactive program in which we can kind of like superficially go through the different ligands, the different chains. Up at the top of this window here, we see the PDB code that we're looking at. If we click on this next, these are clickable boxes. There's really nothing to choose from on this first box, but in the second box here, we can kind of scroll through either the protein itself, the sulfate ion, the APR ligand, adenosine 5-diphosphoriribose, or the water molecules themselves. And this is going to be associated for chain Q. Now, what's going to happen is that we want to go to the ligand itself. And it doesn't matter what chain you're on, just choose the ligand. It's kind of assumed that if you're working with, tetramers or dimers, and they all were co-crystallized with ligands, there's going to be ligands on each of them. But, just choose a ligand on any of the chains to begin with. And then what you're going to do is click on that ligand. And it so happens that, it was kind of like right here in front of us the entire time. When we're looking at this now, kind of went away. Click on the ligand again. We get to like zoom in a bit. What we can do now is move the picture around and rotate things so that we can get a better view of different parts of the ligand and also the protein. Now, what I'm looking for and scanning for here is going to be related to prompt 5. So using the PDB site to find the amino acid of interest. So I'll just kind of leave this window open and then go back to the post lab and just kind of like return back to there because we kind of skipped prompt 4 here. So we finished prompt 3. We chose the ligand. We have the ligand ID and also the ligand name. We'll put that information there. And then now for prompt C4, it says, in the PyMOL program, take a screenshot of your ligand with your enzyme surrounding it. Your ligand should be shown in stick form, and your enzyme can either be shown in line form or in cartoon form. So if you installed PyMOL correctly and you loaded it on your computer, you should have this window that pops up that's going to show 3 pieces of information. So the main one is just going to be this text-based information code that keeps on popping up every time you perform an action. The graphics user interface window here at the middle. And then on the right side is more of like the control panels for executing different actions for what you want to do in the program. PyMOL can be executed in a text-based fashion. Basically, the only way that the only thing that we're going to do this text-based is loading your protein and ligand of interest this way. Everything else is just going to be done through this command center over on the right side because, we don't want to start using codes of line or writing codes of line and there's a bug in it or something like that, and you kind of get stuck just trying to figure out what part of your text-based command is incorrect. So in order for me to load my protein of interest, I'm going to use the fetch command. Again, this is going to be the only text-based command that we are going to be using. Fetch followed by a space, and I'm going to put in my PDB code, 1IHY, and hit enter. All right, so what we can do now is remember that for this particular type of protein that I'm using, the GAPDH molecule that was deposited into PDB, we did see that there was chains A, B, C, and D. And so just by putting the PDB code by itself, I've loaded all 4 of those chains. And I know I've loaded all 4 of those chains because if I click on the sequence button over here on the bottom right corner, I'm shown the sequences of chain A, the sequences. I keep on scrolling through. Chain C was here, so I guess we passed chain B over there on the left. And then, if I scroll a little slower, chain D will show up there. And then all of the other different sulfate ions and ligands in those respective chains. All right, so what I'm gonna do now is we need that picture of our ligand with the protein surrounding it. So it doesn't matter if you take a picture in the chain that you are gonna use in the end, or it's gonna be a different chain. We just wanna see the ligand and then also the protein surrounding it afterwards. So I don't wanna deal with this very complicated tetramer picture either in the foreground or in the background, adding too much stuff that's going to be visually in the way. So all I'm going to do is select here what I forgot to say on the right side for the control panel. Every single object, protein, ligand, the selection that you're currently on has these 5 different commands available. A for action, S for show, H for hide, L for label, and C for color. So if I don't want to close the program out, but I just kind of want to start with a completely new canvas, I can just go to all and select action to delete everything. So when I click delete everything, it's basically like if the program just started from new again. What I want to do now is just load chain A. So I only want to load one part of the tetramer complex. Instead of typing out fetch and my PDB code again, because that was the last line that I used previously, I can just go on my keyboard and press the up arrow while I have my cursor selected inside the command line, and then the previous command will come up there. And all I'm going to do is just fix the capital letter A at the end, and it has to be a capital letter, and that is going to tell the program, all right, this is the PDB code that I want, and this is only the chain that I want to load. So when I click that, I can go ahead and have a single object loaded that is the GAPDH chain A protein. If we did this again but replaced capital A with B, then chain B will show up. Did this again and replace B with C, chain C will show up. And then if we do this again, the 4th and last chain will show up. And because we did it in this fashion, all 4 of those chains can be loaded as their own objects. So you can see these sequences and also their respective sulfate ions and ligands and also associated waters show up and stacked upon each other in the sequences. But we don't want any of this. So all I can just basically do is select, I can select, you can just make them disappear if you want to by hiding them by just clicking on their boxes in the command. But I might as well just go ahead and just delete this whole thing here. Action, delete object, delete object, delete object, because I don't want my right control panel to get too bogged down with too many objects. For these sulfate groups that I have here, you can select them as well and also select the waters as well. And they'll, any selections that you have will show up as its own temporary box, and I could say action, just remove those atoms and they'll go away, so that I'm only left with the enzyme sequence and then also the ligand code itself. When I click on the ligand code, either in action, I can click on zoom, or if you have a 3-button mouse, while APR is the ligand code is selected, you can right click that and then also click on zoom. And this is going to be the fastest way to find and zoom in on that ligand. And basically, this is, pretty much going to be a nice picture worthy type of screenshot that you can take, either by just taking a screenshot of your computer or going to file and export the image as a PNG file. One thing is that I personally like to color my ligand differently than the enzyme itself. So if I have the ligand selected, I can go to color it by element, and the second scheme is personally my favorite because the carbons come out green, oxygens are red, the phosphorus ions are, or the phosphorus atoms, I should say, are orange, and the nitrogens are blue. So take a screenshot of this, and this is pretty much now your figure for prompt C4 in the post-lab write-up. You're already done with 2/3 of the post-lab at this point. Now we have to choose an amino acid and mutate that amino acid, take distances, and also screenshots for that to complete this mutagenesis section. So what I'm going to do is, if you remember from the PDB website, we opened up that one interaction window, and we're given the, all the different chains and all the different ligands associated with that chain. Now it's time to choose an amino acid that we're interested in mutating. And so what I want to do now is look for the ligand in, I just pretty much start at the top of the chains here and see exactly where this ligand is and see if there's any dotted lines that are connecting my ligand to the R groups of the protein itself. Notice that I said R groups and not molecules, because you can have some intramolecular interactions in which the ligand is interacting maybe with the amide group or the carbonyl group on the carbon backbone of the chain. But that's not something that we want to look at for this lab, because we want to mutate an amino acid. It doesn't matter what amino acids you choose to mutate, because if you mutate a tyrosine to a phenylalanine, yeah, the R group changes, but that carbonyl and also the amide group in the peptide bonds will not change, because that is just something that does not mutate as you change the R groups in a amino acid in a polypeptide chain. So for this particular ligand in this chain, I don't see any high potential hydrogen bonds, even though there might be that the computer or the programmer is not capturing here. I just kind of don't want to waste my time too much trying to find something that might not be there. So all I'm going to do is basically stay on the same ligand, but now I'm going to switch from the A chain. So the ligand has the G code onto it, and that's in the A chain. The other APR ligand has the J code associated with it, but it's in the B chain itself. When I select that, nothing happens, and but a new ligand gets presented here in this sequence code here. You can see that in the G code has the 335 number associated with it. If I go to the J code, it has 336. When I click on the 336, now it jumps over to the B chain for this new ligand. I'm just going to kind of survey around here and see what's up. Oh, okay, so over here, I'm spotting the ribose furthest away from the adenine molecule, that it looks like it's not only interacting with the phosphate ion that we really don't care about, but it's got some other interactions there as well. But this doesn't really look promising, because these two oxygens seem to be interacting with the amide group in the peptide bond here. So this is not something that I want to focus my attention on. Moving along. If you have a 3-button mouse, the wheel, the center wheel, allows you to zoom in and out, and right-clicking allows you to pan vertically and also horizontally as well. Going over here on the other side, I see that it looks like, what is this over here? Yeah, this carbonyl group on the peptide backbone is interacting with the adenine in there. So this kind of really is not helping me too much. I'm just going to skip over to the letter P associated ligand 337, click on that and see where I am over here. And I did do this exercise before, so hopefully I can find the group that I'm interested in. It wasn't aspartic acid resin. Oh, I think I found it. Okay, great. So I'm looking over here at the ribose molecule that is over here, the ribose molecule that's closer to the adenine ring. And I can see that these two hydroxyl groups are interacting with what looks like an aspartic acid residue over here. So once we find a amino acid, it's a little difficult to get it to rotate the way you want it to rotate. Once we find an amino acid residue that looks like it's participating in some hydrogen bond interactions with your ligand, now what you want to do is just carefully click on that amino acid, and you're going to get information that's going to be helpful for you. Jesus, clicking too much does weird things in this program, which really annoys me. So yeah, zooming in again on this aspartic acid residue, I'm just going to click it once. And here, over on the right side, I get some information as to which amino acid that was, and then also which chain is associated with it, and also the number itself. So this was, we want to go and focus on aspartate 31 in chain D. So I'm going to jot that down, go back to the PyMOL program, and again, this was just, what we have here. It was getting a picture of the ligand and the protein. I just kind of want to start off with a blank canvas now and then delete the selection, since this is chain A and we actually want chain D. Sorry, delete everything, not delete selection. I'll go back to the fetch command and make sure that I load chain D. So 1IHY, fetch space 1IHY, and then a capital D. Remember that the chain letters need to be capitalized. When I click that, now I have my protein and my ligand loaded up there. I'm just going to really quickly take those sulfates and that water, and remove those atoms so that I'm only left with the sequence and also the ligand code of my protein. I am going to then click on the ligand code up in the sequence scroll bar and right click that and select zoom. And now what I'm looking for in the sequence is aspartic acid 31. So here I have, or I think it was like 32 or something, but anyway, it's this one right here. So it's the letter D. I'll select it, and now I can see, one thing about selecting is that it's really weird. You don't need to have the, press shift or hold it down. Anything that you are selecting continues to get selected and grouped. And so this is kind of a little annoying, especially if you don't realize this, perform a command, and then now you've done something that, looks a little messy. So to unselect, essentially just click on some black space here in the graphics user interface panel. So I'm going to click on aspartic acid 30, I think this was 32. And I'll see here that this is the amino acid that pops up over here. Now, currently it is not being shown, it is hidden. By default, all the amino acid residue groups are hidden. In order to show them, I'll just go to show and show as sticks, and then also color those using my second element coloring scheme that I like. I don't recommend that you start showing too many amino acid residues, because then it's just going to get visually complicated, um, and so on and so forth. So I'm working currently on prompt 5, and it wants me to capture the wild type protein and the ligand and a hydrogen bond. And so what I'm going to do is, and I remember that it was the aspartic acid and the ribose between the diphosphate and also the adenine group. In order to capture these hydrogen bond interactions, we need to see the hydrogens. So for both the ligand and also the currently selected amino acid residue, what I'm going to do is, for the amino acid residue, it's under the selection because I currently have that selected. I'm going to go to A for action and then click on hydrogens, and another submenu pops up, and I want to say that I want to add the hydrogen. So if there's any hydrogen bonds, or sorry, any hydrogens that are associated with that selection or that molecule, they'll pop up as these little gray sticks. For my ligand, I'm going to go find the code, the ligand code in the sequence bar, and click on it, and also go to action for the selection because it's currently the only thing being selected, hydrogen, and add. Now, the reason why I like that second color scheme is because now the carbons look like hydrogens, and that just visually is a little confusing for me. So while I still have the ligand selected, I'll go to color by element, and I'll choose the second elemental coloring scheme, which now differentiates the carbon, the green carbons, from the gray hydrogens themselves. All right, so we're almost done with prompt 5, except we need to document a hydrogen bond distance. And so I think if we look back at the interaction 3D diagram program, this deprotonated oxygen off of the carboxylic group in aspartic acid looked like it was interacting between these two hydrogens here. So I'll just focus on one of the hydrogens in the hydroxyl groups in the ribose sugar itself, and I'll just choose this one, the one that's closest to us, closer to the diphosphate groups rather than the one that's closer to the adenine ring. To make a measurement, we need to go to the wizard toolbox or the wizard command set up at the top of the program. Click on wizard, select measurement, and now we are in the measurement wizard interface itself. And we know we are because a new command comes up on the left side, and then on the, closer to the bottom right, we have this measurement dialog that pops up. The command says click on the first atom. So what we're going to do is, I want to zoom in a little bit because I don't want to accidentally click something that I'm not supposed to. Click on this hydrogen here at the end of this hydroxyl group. And then now it says, please click on the second atom. And I'm going to click on the deprotonated oxygen in that carboxylic acid group in aspartic acid. And now the measurement that shows up is 3.5 angstroms. Now, please be careful when you're in these wizards programs, because you are still stuck in the wizard program until you click on done. When students, forget that they're in all these like wizard measurements and stuff like that, they keep on clicking everywhere, and they're really confused as to why they're getting measurements everywhere when they don't want them or didn't ask for them. That's because you're still stuck in the wizard. So click done, and if you do have any measurements that are unwanted and do pop up, you can just go ahead and click on those measurement entry boxes under action, and then just delete them one by one without having to rage quit and start the program all over again. All right, so this is great. This is a nice hydrogen bond distance. I'll take a screenshot of this. If you are having trouble finding one between the 2.5 and 4 angstrom recommendation, if it's a little under, a little over, that's fine. No, that's not really the scope of this post lab to find something that's perfectly within that range. The larger goal of this post lab is to mutate an amino acid and change that hydrogen bond distance. All right, so after we're done documenting this, now we are going to move on to the last prompt in the post lab, which is prompt C6. So now it says mutate your amino acid of interest to change the hydrogen bond distance. Notice that I said change because we were having a little bit of trouble of students finding amino acids that would either, that should theoretically decrease the hydrogen bond distance to strengthen the interaction. But, we're a little lenient. We're just saying, as long as you change the hydrogen bond distance, you either increase or decrease the interaction. We don't actually know how to quantify that stuff without actually doing proper experiments. Then, any change is fine. Measure the hydrogen bond distance between the mutated amino acid and ligand. Take a screenshot, and ligand should be in stick form, and the mutated amino acid should also be in stick form as well. All right, so what we are going to do now is I'm just going to hide this measurement by clicking on the object entry itself, and then I'm going to go to the wizard, and under mutagenesis, I can choose either to mutate a protein or mutate nucleic acids. Now, this nucleic acid thing, it's actually pretty new. I think it's new within like the past 2 years as a transcription factor, biologist in my graduate work. I freaked out when I saw this nucleic acids thing. Basically, for any transcription factors that have been deposited in PDB, now you can kind of like mutate the DNA that would be in the ligand surrounding these transcription factors, or also like restriction enzymes. And it was actually pretty cool to do, especially now that like CRISPR is so big and hot in the field. But we want to mutate the protein and amino acids. So we're going to choose mutagenesis and protein. And now, again, we're in a wizard. There are some commands associated with it, so be careful. The first thing it says is pick a residue. So the residue that I'm going to pick is that aspartic acid residue 32 there. And when I select it, you see it's weird. Things kind of like pop up and shift and stuff like that. So what we're first presented with is kind of like a little clone of that amino acid residue, and it's going to be in a slightly different conformation known as a rotamer. And right now we're in rotamer state 2 out of 3, meaning that there are 3 total different spatial configurations that this R group can be situated in the program itself. And if we scroll through these different rotamers using the backwards and forwards button, be careful, don't press this like play button, because if you press it, then the rotamers just cycles through all the rotamers and it just looks really scary and crazy. Press the forward button or the backwards button to kind of rotate through all of these rotamers, and which is something that you might want to do with your newly mutated residue. But after you selected the amino acid that you want to mutate, you can change the rotamer, or you can actually change the R group itself. So up here where it says no mutation, that was because it was probably selected that no change should happen. But we want to change this, or I want to change this from an aspartic acid to a glutamic acid, or a glutamate. The aspartic acid that is here in the program, it has a deprotonated carboxylic acid group, and so I am also going to choose the glutamate with a deprotonated in the deprotonated state, as indicated with this missing H here. So when I click on that gray clone ghost basically changes into the glutamate, which is 1 carbon longer. And now we can see that we are presented with 6 different rotamer conformation possibilities, and we're currently seeing the 4th one here. To me, this just looks great and it looks perfect. So you can see the old aspartic acid residue there at the bottom. Really, if you hold down shift and you or maybe control, if you hold down control, or I think it's command on Apple, and then drag your mouse, you can also kind of move laterally, horizontally, and vertically. We can see that the new glutamic acid residue is going to be much closer in proximity to that hydroxyl group off of the ribose molecule, in this rotamer conformation. If we want to, change the rotamer conformation and see if we can get something that's a little bit closer, great, that's fine, go for it. But just be careful. If anyone's wondering, these, short red cylinder discs that pop up, this is just kind of showing you molecular strain. And there is some quantifications associated with this that comes up, but we're not having you jot down any of these or do anything with this for this lab in and of itself. Once you're done choosing your desired mutation, then click apply, and just the apply step will remove the old amino acid residue. It'll make your mutation happen, and then the wizard starts all over again, because over on the upper left side, it's saying pick a residue. So you're still stuck in the wizard in case you want to do another mutation, that's fine. But I personally am done with this, so I'm going to go select done in this wizard dialog box here and just get out of the wizard before I start mutating the whole darn protein. All right, last step that I need to do is just basically do a measurement. So I'll go to wizard, measurement. Again, click on the first atom, that same hydrogen off of the hydroxyl group in the ribose sugar, and that deprotonated oxygen now in the carboxylic acid group in the glutamate residue gives me a angstrom measurement of 2.4, which if I go back and make the first measurement appear by clicking that entry box, I see that it is about, what is that? 11 angst, or sorry, 1.1 angstroms shorter than the first measurement that I had there. But for the purposes of prompt C6, now basically all we want to do is just, have the ligand in view, have that mutated amino acid also in view, and then that new angstrom measurement. Take a screenshot of this, don't forget to exit the measurement wizard, and then complete the post lab by finishing this prompt here. Some students have been kind of like wondering, well, what should we write for the conclusions? Because there was actually no experimentation. Yeah, that's right. There's really nothing that we did experimentally here. We did propose some changes to either strengthen or weaken the interaction between the ligand and the protein, but kind of tie this into current research that's happening right now. In the field of medicinal chemistry, these types of studies are very, very important because on the other aspect, they're not really mutating proteins, but they're, quote unquote, mutating the ligands. They are changing and adding different functional groups on currently existing drugs to make drugs better, to start killing off, antibiotic resistant bacteria and cancers and kinases and things like this. So you could talk a little bit about that, but for the most part, just 4 to 5 sentences in the conclusions about what you learned with this and how this can be applicable to modern medicine or modern science in the fields today. All right, so if you guys have any questions, please email me, let me know, and hopefully everyone gets a chance to finish this post lab before it's due. All right, thanks everybody.